So very good afternoon to all. So I'm basically here to present my work on Graphene-Oxide-Assisted Biosensor with optimum response selection algorithm for detecting and quantifying Vimentin, a potential biomarker for ovarian cancer. I'm Ullas Pandey and I'm from IIT Hyderabad. Yes, thank you. So our focus of our work is to develop a portable, cheaper biosensor which can detect ovarian cancer in early stages. Since in country like India, where among gynecological malignancy, the highest death rate is caused by ovarian cancer. So our main focus is to develop a cheaper biosensor which can be used by general public, like glucometers, and it does not require very medical facilities or very high equipments. So regarding this, we have as you know that the available techniques for detecting the ovarian cancer is ultrasounds, biopsy, CA-125 antigen detections, CT scans, and genetic tests. So what we have done that we have taken out a panel of biomarkers, and among which few are FDA approved also, and we tried to detect these biomarkers in the blood plasma. So among these proteins, one of the important biomarkers which we have found is Vimentin, which is a type 3 intermediate filament protein expressed in mesenchymal cells. It's a kind of structural protein which is responsible for maintaining cells integrity, shape, cell migration, and adhesion. It is also a canonical marker for EMT phenotype. But this protein gets upregulated in ovarian cancer, and also it is expressed in the cytoplasm and plasma membrane of ovarian cancer cells. It means that it is there, it is present there in the plasma of patient, and if you could detect this in the plasma, then we can early detect the ovarian cancer. So the basic mechanism of this biosensor is the chemiresistive technique, where we have used fabricated a sensor chip which consists of interdigitated transducers. It is fabricated on PCB substrate, and also which is electroplated with a 100 nanometer gold layer. We have used graphene oxide as a sensing material to develop this biosensor, which is a very well-known 2D materials. And you can see the same images of the geo-decorated IDEs in the screen. Also, we have done the TM images of the graphene oxide, and we have done the IDEs spectrum, that is the elemental mapping of the graphene oxide. So the basic technique for this sensor is the chemiresistive technique, and it describes this describes the whole scheme of the preparation of the biosensors. So first we broadcast well-formed dispersion of geo on the sensor. Then by using P-based treatment, which is a linker, we immobilize the antibodies on the sensors, and then we do BSA treatment to block the sites which are not covered by the antibodies, and then we expose our sensors to the plasma samples which contains the antigen of the Vimentin protein. And this is the whole scheme. After that, we measure the impedances. We carry out the IV characteristics, and then we study that what actually happens when antibody antigen binding takes place. You can see that in the figure 1, that as we since these are antibody antigens are biomarkers, and so as they combine, as they interact with each other, it increases the impedances of the device. You can see that after BSA treatment, when we expose the sensors to the plasma, and plasma contains the Vimentin protein, the current decreases, and it goes on decreasing in the same fashion as we increases the concentration of antigen in the plasma. So you can also see that when we see about the resistance versus voltage response, then we can see that since graphene oxide is a very conductive material, so the current is very high and resistance is very low. But as the antibody is immobilized on the surface of the sensors, the resistance enhances, and as we do BSA treatment, also the sensor the impedance enhances. After combination of after it is exposed to antigens of the Vimentin, the resistance is further enhanced, as you can see in the figure 3. So since in this case we have taken voltage from 0 to 1.5 volt, having a step of 10 millivolts. So the basic problem is that our sensors can have high response at any voltage between 0 to 1.5 volt. Our main aim is to detect that at what point of voltage a sensor have the optimum response. For this, we have developed an optimum response selection algorithm, which is coded in Python. So what it does, first it takes all the waveforms of the different devices, and first it smoothens the waveform. After that, it adds all the waveforms, and then it do a scan throughout the voltage points. So what we have done, we have taken for the scan, we have taken points from 25 to 35 voltage points, and we have done scanning from. So we can see that the scan goes like this: that it is start from the voltage point 0 to next 23 points. Again, it will start from voltage point 1 and next to 23 points. In this way, it scans, and it does this scans for different points. That like it will start from 25. It will first take 25 points, then it will take 26 points, then it will take 27 points. Like this, it will go up to 35 points, and it will check that for what scan, for what number of points we have the optimum response. And then it comes and backtrace that at what voltage it was giving the optimum response. So in this case, you can see that for 26 points scan, we get the highest optimum response, which is between 1.41 and 1.42. And then we backtrace the voltage, and we found that between 0.53 to 0.78 volt, we get the highest response. In this way, we have taken the response of our sensors, which is better than the earlier techniques where people generally used to take the average of all the responses over a voltage interval. So when we plot the calibration curves, we found that we can see that in the first figure, that by using ORSA algorithms, the steepness of the calibration curve has increased, and that's why the sensitivity has got enhanced, as well as we have got a lower LOD with respect to the others normal techniques. We have also done we have also taken the response of our sensor for different interfering proteins, since the clinical samples contains hundreds of other proteins. So the biosensor should be as selective for the respective protein. So in this case, we have studied the we have done the interference studies with different proteins like fibronectin, troponin 1, STRM1, TNF alpha, and we have also made a cocktail of all these proteins and taken the IV responses. And we can see that the response for our biosensor for this protein, Vimentin, is far better and far than for the other proteins. So I'll conclude here that we have done a broad response for target antigen concentrations from 1.28 picogram per ml to 8 microgram per ml concentrations. So by using Python-based ORSA, we have boosted the sensitivity of the device and lowers the LOD. The given sensitivity is 19.528 kilo-ohm per microgram per ml per unit area of the sensor, and LOD is 0.956 picogram per ml. So, and selectivity and interference studies showed that there are towing potential for biosensors for clinical sample tests. For the future scope, I can say that if you could make a readout for this sensor and if you could integrate it with microfluidics, then it can be used as a point of care diagnosis in the real applications. And as for now, we have already developed the microfluidics channels to separate the plasma from the blood, the whole blood. And also we have developed till now that we have developed portable readouts. So we need to integrate this device so that it can be used as a point of care diagnosis. Thank you.